Hello, this is Patrick Sullivan, and I'm one of the principal investigators of the PGC and one of the co-founders of the PGC. And I'll be talking today about the future perspectives of the PGC. I have a couple of conflicts to disclose. I've been a consultant and a grantee for Lundbeck, a pharmaceutical company, and I'm on the scientific advisory board, and I'm a shareholder of RB&C Therapeutics. So one of the key advantages of the Cytogenomics Consortium in the approximately 12, 13 years that we've been in existence is that we've brought sustained attention by a large number of investigators worldwide to psychiatric genomics. This has been astonishing, and to my knowledge, it's unprecedented. What I'm showing here, each of the dots is a place that had a person who co-authored a PGC paper. And the arcs, the faint yellow arcs, connect them. And as you can see, we've had a huge number of people from the United States, mostly Western Europe and Australia, to be have been part of the PGC. And clearly this is great and it's fantastic, but it's also not complete. And if we look at the globe, and if we look at the places in the PGC, for the countries from which we've gotten cases, what we're looking here is coloring by country. It's on a log 10 scale, so 2 means 100 and 5 means 10,000. Sorry, 5 means 100,000. As you can see, the most cases and controls that we've gotten have been from Western European countries, as well as the US and Australia, and certainly an increasing number from China and other countries in East Asia. But as you can see, all the way across sort of an equatorial band, Tropic of Cancer to Tropic of Capricorn, we've done a fairly poor job. And that has to become something that the PGC actually works on heavily. And I'll talk about that later in this talk, but it's a key issue for us. We've actually published on this. There was a leading edge primer in Cell on this topic. And Mark Daly, Ben Neal, and Alicia Martin and colleagues actually had a really interesting paper on how this can actually lead secondarily to some health disparities. But one of the things we've learned in the 12, 13 years that we've been doing this is that what we've discovered is actually different from what had been assumed previously. So if we look at what we know right now compared to what people thought was the case in 2005, a lot of things are different. Candidate genes have rarely stood the test of time. There's a couple of papers that have formally looked at this for schizophrenia and for major depression, and what we thought was the case is generally not. We've done an awful lot of looking to see whether we can find rare variants of very strong effect that are causal. And to be honest, there's not a lot of there. There certainly may be some for autism and intellectual disability, but for the adult onset psychiatric disorders, they're few and far between. And there's certainly examples in human genetics, Alzheimer's disease, breast cancer, where rare variants of strong effect can, in fact, explain small proportions of cases. But these really are truly complex traits. Tens to hundreds, if not thousands, of common variants are involved. And really, this covers virtually all traits that we've studied to date, including MRI volumetric studies. And if we look at, this is from a paper we've written recently about a field review looking at technological readiness in a clinical setting. And so we've looked at multiple different areas of medicine, from allergy immunology all the way down through psychiatry and autoimmune diseases. And essentially what we see is that for most conditions, occasionally there's some rare variants of strong effect that are diagnostically important. For example, the familial conditions, if somebody has a certain type of heart disease that strongly runs in families, there's established tests. For monogenic diabetes or very early onset diabetes, for instance, certainly that too. Alzheimer's disease, definitely, et cetera. Some of these can actually impact treatment, not many. But then if you look at where we are across many other areas, including psychiatry, so typically there are a few things that one can do right now for diagnostic purposes, a few that may be relevant for treatment, but it's not clear, and then for everything else is still exploratory. So the level of readiness here, 1 means essentially that it's investigational. 2 means that we could probably do a trial if there's good evidence for that. and 3 means it's actually in widespread clinical practice. So psychiatry is actually not that different from any other field, with the possible exception that we don't have proven strong variants of that are of clinical and of diagnostic relevance. Now, in terms of in the years the PGC has been in existence, we've published 320 papers as of the end of 2019. As you can see, there have been a marked uptick in the number of papers in 2018 and 2019. This is because of our policies about secondary analysis, where it's actually relatively easy for any PGC investigator to write a paper using PGC data. And the PGC has also been quite impactful. So this is from the NIH iCite tool, where they take the number of citations that a particular paper has compared with what you would expect for a grant from the same US National Institute of Health Institute, as well as the same topic area. And they scale it so that the median citation is at 50, and the median PGC paper, this is a graph of all the PGC papers and the citation index for each of them on the scale. The median of the PGC papers comes in on the 85th percentile. So a typical paper is really much, much better than the median across NIMH. And the top paper, the 2014 schizophrenia paper, is one of the most highly cited papers the NIMH has ever had. We've had 3 papers in Cell, 1 in Lancet, 3 in Nature, and multiple papers in Nature family journals, and 2 in Science. So 320 papers. The PGC as an author, so to speak, has an H index of 65, mean of 92 cites per paper, and 59 of the papers are actually listed as being highly cited in Web of Science. So the PGC really has been quite productive and quite impactful. And one of the key things, though, is how data-driven we are. That's really been something that's been baked into the DNA of the PGC from the very start. We're empirical. We will test ideas in the field. We will not take them for granted, and we believe in the data. And we don't publish things until we've tried again and again to disprove them, and then we go ahead with all the appropriate cautions just to make sure we're coming up with lasting knowledge. So what I'm showing here on the left side is the structure of the PGC. There's a coordinating committee. There are working groups for the different disorders, ADHD, Alzheimer's, anxiety disorders, et cetera. We've got a new trans ancestry group coming up, the data access and outreach group, and there's also a dissemination and outreach committee. The ones that are in gray were the original groups, so ADHD, autism, bipolar disorder, CNVs, cross disorder, depression, and schizophrenia. And the ones that are greenish are the somewhat newer ones, mostly within the last 3 to 5 years. The trans ancestry working group is one that we're launching should the PGC4 grant get funded. And in terms of just the state of play, this is taken from a slide by TJ Singh in the schema paper that's available on medRxiv, I believe, and will be resubmitted shortly. What it's showing here is the minor allele frequency in the general population on a log 10 scale and the odds ratio on a log 10 scale. And what you can see is that these are the common variants that have been discovered of relatively strong effect. SLC 39A8 looks pretty good. C4, I would argue less so, but these are the findings that have come from GWAS. Over here, you can start to see things that are relatively rare, but relatively strong. And the ones in green are copy number variants that have come up. And the red ones are ones that came up in an exome sequencing study, the one here, schema. If you do a search for Broad Institute and schema, you can find this page and you can look at all the genes that have been implicated. So what we see, in fact, in terms for the genetic architecture of schizophrenia, and that's what this slide is about, is essentially a mix. Most things are common variants of soft effect, but occasionally you find rare variants of stronger effect. However, many of these are awfully rare. One of the strongest genes actually is SETD1A, and I believe you find a variant, an impactful variant in that, an exon variant in that gene, maybe in one case in 1,000, and effectively 0 controls. That's the kind of signal, and clearly it's strong, but it doesn't explain a lot of cases, although it does give us a nice inroad into the biology of the disease. And then There's been a lot written about the so-called missing heritability, and it's quite likely that as we move to whole genome sequencing, a lot of this will be recovered. Peter Vischer and colleagues had a paper where they actually did this for height and BMI using the top med whole genome sequencing data, and were able to recover most, if not all, of the missing heritability when they got into variants that were somewhat uncommon and not well tagged by linkage disequilibrium. These are some of the highlights of the PGC. This is just sort of the title slides for a bunch of the Nature Genetics, Nature Neuroscience, and Cell papers. One that I particularly liked was the cover that our substance abuse colleagues got in Nature Neuroscience, where they had this sudsy Manhattan plot. So what I'm going to talk about now is our plans for the 4th PGC grant. So this has been, we put in the application in February. We received a fundable score from the NIH. We've had promising comments and conversations with people at the NIH, but it is certainly not yet for certain. And we never, ever take this for certain until we actually get the notice of award. So we're hopeful, but by no means is it certain. But let me lay out for you what we discussed in the grant. So there were 5 aims. The first one was essentially the core business of the PGC, continuing in the process of genetic discovery. So what we've been doing has been quite useful and informative, and it makes no sense to not, we need to keep doing the same. And this is what this is about. We want to make sure that we know how to carefully get in the large biobank studies into the PGC analyses. But one of the key parts of this aim is to increase the ancestral diversity. As I mentioned before, this is a big part of that. The PGC so far really is mostly European in nature, and we urgently need to get a lot more of humanity involved in the PGC. We need to have studies from India. We need to have more studies from East Asia, definitely more studies from Africa, and certainly Latin, Central America, Mexico, and the Caribbean nations are also places that we certainly need to get more information from. And just to give an indication of how important this is to us, it was one of the first things I mentioned in this talk, and it's actually part of our first aim. And I guess one of the reasons that was one of the really cool thing from the PGC paper that Haile Lang and Max Lamb led that came out last year was this was a large GWAS of East Asia and European populations, and the genetic correlation for schizophrenia between the two populations was essentially 1. The genetic correlation was 0.98 with a confidence interval that easily covers 1. It's the same condition. So schizophrenia in East Asia from a genetic perspective is essentially identical. The common variant basis of it is essentially identical to that in European nations. All the more reason to do this. We need to extend, we need to bring in lots of other samples and more data for everyone's benefit. Our projections in terms of what's going to happen and where we are. This graph shows the number of cases and the number of associations for each of the PGC disorders. Schizophrenia has been submitted and will be resubmitted soon. It's at 270 genome-wide associations in about 67,000 cases. Bipolar disorder is well over 50. I believe the most recent analysis is upwards of 100. Depression is over 100 in terms of what's published. And if you want to see some cool stuff, listen to some of the talks by Andrew McIntosh and Catherine Lewis for where it is now. But they've really managed to markedly increase. I think most disorders actually have at least 1 finding these days. And really, we need to get all of these conditions on the slope to discovery. In terms of the current sample sizes, they're listed here for each of the disorders. These are the number of cases. But as you can see, the proportion of individuals that are not of European ancestry is far too often 0. Schizophrenia has done a relatively good job, PTSD, opiates, the substance use disorder, et cetera. But everybody needs to do better. The current number of loci are listed here, and these are the data that are plotted on the graph. And these are what the group leaders project in terms of the sample sizes in 5 years' time. And so we expect to have over 2.5 million cases with GWAS data in the PGC in 2025. Believe it or not, this isn't enough. We need more. And that's one of the reasons I'll have some more comments about that later in the talk. And we asked the PGC group chairs when they made these estimates to please do it in a relatively conservative way. And so I think this is actually super encouraging, and this is something that we can definitely do better than. Okay, so again, coming back to this, we really, really need to improve our coverage of other groups in the world. And the one thing I want to mention is we really wish to extend an open invitation for anyone on this call to become part of the PGC. We're aware that there's a lot of people who are coming, who are at this meeting this time or watching this offline, as the case might be. Way more than we typically get at this conference. And again, I would like to extend an open invitation for you and your colleagues to become part of the PGC. To become part of the PGC, one needs samples. It's not open per se. It's open to people that can bring data to the table. Typically, in the past, it was that we had to have GWAS data in hand to become part of the PGC. I think we should change that. And if somebody has a sizable sample of DNA that meets, decent criteria for diagnosis and ascertainment, and if the ethics are online, I think that should be enough to become part of the PGC. So I would suggest this. My 2 cents are, lay down your own sample of cases and controls. Many people on this call, many clinicians have access to very large samples. And if they can get 1,000 cases and 1,000 controls, we can probably get them genotyped somehow. At the same time, though, when you do this, it's important there's a number of things you have to think ahead about. And I would advise you to contact the PGC group that you're interested in, bipolar, schizophrenia, whatever. If you go to the PGC site, you can find the group leader. If not, shoot me an email and I'll connect you. But the basic idea is contact the group for advice about the diagnostic instruments to use. We accept a range of things, but it has to be done in a way that's comparable with everything else. It's always worth thinking ahead about ethics and informed consent, as well as the type of DNA that you lay down and its nature. But typically, blood is great. Many people who work in clinical samples can get an extra tube of blood. It has to be stored in a certain way, but that usually isn't too difficult to do. But really, this must be a win-win for everybody in the end. Our goal, at least the goal I've had in a number of the groups I've worked with in non-European countries, is the explicit goal in the memo of understanding we have is for our colleagues to be able to do independent world-class cytogenomics. We want to get to that point. It takes time, it takes effort, but we can help get people up to the point where they can do independent work by themselves. And I think that really is something that we need to have as sort of an end goal for this. Okay, that was aim 1. Aim 2 really is advancing discovery across the genetic architecture. So again, we have a lot of stuff going on the common variant side. That's what aim 1 is all about. But at the same time, we can always we can look at rare variants in a couple of different ways. So, and we have 3 different parts to the same. The first is the PGC CNV group, copy number variation group. We want to make sure that we can do all the disorders in a very systematic way. That will enable the CNV analyses across diagnostic groups, which I think is the next thing we really, really need to do. In addition, Aidan Corvin and colleagues have been doing whole genome sequencing in densely affected pedigrees that have been sampled across the world. The goal there is that these might be pedigrees in which rare variants of strong effects might be segregating. I think mostly they're going to be just common variation that tends to pile up in a family. If you have 2 parents that are standard deviation above the mean, that will translate to increased risk in the offspring of a dense family. So it can be for common variant reasons as well, but it might be for rare variant reasons. And I think we need to do a much better job at going after families like that. And Aidan and his colleagues have a great pipeline, and they've done a lot of work, and there's a bunch of papers coming. And finally, I think a really, really interesting aim is that most studies do genomics on their sample. They write a common variant GWAS paper, they write a rare CNV paper, and then they write a whole exome paper. And those 3 things tend to occur in isolation. And clearly, those are papers that are organized around a technology, and that doesn't make sense. It should be organized about the disorder. And so there's Mark Daly, Ben Neal, and others will be actually doing a lot of method development in this area, but it's really, really important to figure out how we can actually, in the same study, integrate and look at all these different types of variation in the same person so we can get a far better idea of the causality factors in a particular individual. The third aim is to really look carefully at our standard diagnostic instruments. And so there's a couple of parts to that. The first is that we need to do a much better job at phenotyping. We have to have large samples that actually have the same phenotypes. And the reason why is it's been really difficult to harmonize instrumentation after the fact. And if we do it ahead of time, especially if we have a lot of new samples coming in, that would actually be very, very useful to make these kind of samples, make this kind of analysis instantly possible. In addition, I think the PGC can actually, is at the point where we can go back and start to focus on some critical questions of clinical relevance. So for example, can we identify subsets of patients using genetic data? No one would think these days that schizophrenia, people with schizophrenia are all the same. Can we use genetic data to try and uncover the heterogeneity within that? What about therapeutic outcomes? Can we use genetic tests to help us understand who might have a good versus poor outcome? The other thing is, how do genetic effects vary across the lifespan? What happens if there's a real strong stressor? And unfortunately, the COVID-19 pandemic has given us a terrible but potentially useful scientific way to look at this. Who are the people that do well versus poorly, given the stressor of the pandemic? And then the one thing the PGC can do that I don't think anyone else can is we can use our global network to engender new studies of highly severe or treatment-resistant cases. And in this way, enrich causal alleles and also increase the translational potential. And so here are just a few examples of different people in the PGC that have done this. We have a study called Mom Genes with Sam Meltzer-Brody and Jerry Quintavano at UNC. And essentially we focus on postpartum depression. We had previously shown that it's a more heritable subform of depression. Jerry and the team are finishing up a GWAS of 17,000 cases right now. Our colleagues in Cardiff have a study called CLAS UK, where it's based around people with schizophrenia getting clozapine, so clearly, treatment resistant. We have a study in Pennsylvania called PASH, where we have 500 people in the study who were ultra treatment resistant. These people had to be hospitalized, actively treated for at least 5 years and still have substantial symptoms. Peter Zandi and I have an NIH grant for the genetics of ECT. So we've shown that the SNP heritability of people getting ECT is much higher. They have higher GRS burden. And so Peter and I levied this into a grant where our goal is to get and genotype 25,000 cases. And our PGC colleague, Bernard Bauna, actually there's a PGC MDD subgroup or interest group on this topic too. So that's one example of something that the PGC can do that would be difficult for others to do. And finally, our colleagues in the anorexia nervosa group have managed to find a sample of people with very severe enduring anorexia. These are individuals who've been ill for 30 years, more or less continuously, treated many, many, many times, and still are quite badly affected. Aim 4 has to do with trying to uncover novel therapeutic and preventative opportunities. So here, something that's been in the PGC papers for a very long time is try to understand the biological consequences. A number of different ways you can do this. There's a bunch of talks on this there were a bunch of talks at this conference. We're going to be working closely with PsycEncode on this. In addition, causal risk factors can be delineated using a new technique called Mendelian randomization. A bit too long to explain here, but if you've got an epidemiological risk factor and you don't know the direction of causation, MR can give you a hint as to whether it's causal versus reverse causal. And the final thing is to combine the epi and polygenic modeling to try to see whether these models can actually help us understand patient stratification better. And then finally, aim 5 is to increase the impact of the PGC by making sure we're talking to the right people in the right communities. And so we will continue our work with medicine discovery groups. Jerome Breen has had 3 PGC/pharma meetings where we've tried to help them understand what we're learning and to understand the ways in which it might and might not be useful. The PGC Outreach Committee is in the process of using digital media to make sure our findings are getting communicated in an accurate and real-time way. We're in the process of creating some educational resources for patients, families, and medical professionals, especially in regard to the testing, testing for rare variants and seeing these in various patient groups. And then final, we're going to establish a scientific advisory board, which we've never had before, to make sure that we hear the concerns of a wide swath of our end users, and to make sure that we have maximized PGC alignment with with those groups. Okay, so I think the schizophrenia, bipolar disorder, and major depression are arguably at the end of the opening phase, at the start of the middle game. Other groups are still not quite to that point, but at the same time, the findings here have shown us what can occur if the sample sizes get large enough. But again, expansion to non-European samples is absolutely critical for us. And I think we really need to particularly get severe cases. There's a bunch of opportunities for depression and ECT and schizophrenia, schizoaffective disorder, and bipolar 1. A lot of us are actively looking for postdocs. And so if you've got a clever grad student who you think would be interested in this, please let various people know. And then of course, the final thing is there's a bunch of opportunities to help us upskill people. Again, I think the ideal outcome of getting other people involved is that everybody would learn a lot more about how to do genomics and eventually get to the point where they can do it themselves. Okay, that's it for my talk. And I thank you for your attention. And I look forward to taking some questions.